As you know, we have completed protein synthesis in 10 lectures. Now we are going to demonstrate it here on smartboard. Please try to learn things very clearly. This will make you fully understand this topic, protein synthesis. So please be careful. Sound बढ़ाइए. Sound बढ़ाइए. You guys क्या विषय दिया? Sound बढ़ाइए भी. हाँ.
The Bluetooth device is connected successfully.
Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones.
As per your DNA packaging.
Yes, sir.
How DNA is supercoiled inside nucleus. Supercoiling. Do you remember supercoiling?
Yes, sir.
How chromosomes are formed? DNA is wrapped around nucleosomes. Nucleosome is made up of which proteins? Histone proteins. एक न्यूक्लियोसोम में कितने हिस्टोन प्रोटीन्स होते हैं? Because this is also called octamer. 8 हिस्टोन प्रोटीन्स. कहा था समझते? This is हिस्टोन. चलो. और ये इसको अनकोइल करेगा, हाँ, अनकोइल करके दिखाएगा, क्रोमोसोम को अनकोइल करेगा और डीएनए निकलेगा. चलो. What happened?
Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene.
You got to check.
Yes, sir.
Which process?
Transcription.
Transcription. Formation of RNA from DNA. If you go RNA polymerase. You
It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription.
So mRNA is formed
Yes, sir.
from DNA.
Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed.
Processing. You guys got saw processing कैसे?
Yes, sir.
चला आपने ना introns exons आसन इसमें कोई नहीं होता है.
Yes, sir.
अब इसकी process हो जाएगी पहले.
This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the
Two nuclear poles.
cytoplasm.
आ गया बाहर cytoplasm. आया? गया cytoplasm. Message
Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids.
What is this? What is this? tRNA molecule. आपकी बॉल के एंटीकोड आ रही है. Three bases. In this क्या लोड है? What is this? Amino acid. The first amino acid.
AUG.
First amino acid. Methionine. Oh, that's methionine. So in every protein chain, the first amino acid is methionine. Which is methionine in the load tRNA molecules. Here it will come. Will go to smaller subunit on in which site? पहले सब
P side.
P side में क्या? P side.
There are 20 different types of amino acid.
Are you waiting?
Yes, sir.
Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids.
ये chain पे grow कैसे? Protein chain, amino acid chain, है ना?
Yes, sir.
Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
अब इससे आगे जो वीडियो है वो सबसे अच्छा है, वो आएगा. अब देखना.
Protein synthesis translation.
Inside the body, the process of translation occurs within every single cell. Each cell has a nucleus. After transcription, mRNAs move out of the nucleus and enter the cytoplasm. This mRNA strand acts as a template for protein synthesis. Present in the cytoplasm is an enzyme, amino acid tRNA synthetase. The enzyme macromolecule has two binding sites. One site recognizes the amino acid methionine. This is followed by the binding of the ATP molecule
Charging up here.
and release of pyruphate,
tRNA molecule.
resulting in activation of amino acid. Finally, the tRNA and the activated amino acid bind together. This amino acylated tRNA is known as met-tRNA
Met.
and is released from
Why
the
it is called a met tRNA? Because it is loaded with methionine amino acid. So it is called, you should miss methionine, it's called a met.
enzyme. This marks the commencement of first stage of protein synthesis, the initiation stage.
Initiation stage.
During the initiation stage, a small subunit of a ribosome binds to the mRNA strand.
Wait.
The mRNA strand is made up of codons, which are sequences of 3 bases.
Codons. These are codons.
Then the ribosome subunit moves along the mRNA in 5' to 3' direction until it recognizes the AUG codon or the initiation codon.
ये AUG codon, first codon. Initiation codon.
At this point, met-tRNA possessing the anticodon UAC pairs up with the AUG codon of the mRNA.
You just pay attention.
Then a large subunit of ribosome combines with a small ribosomal subunit. The large subunit shows 3 sites: the acceptor site or the A site, the peptidyl site or the P site, the exit site or the E site.
जो अब पहला tRNA मॉलिक्यूल आएगा विद एमिनो एसिड मिथ्योनिन चार्ज विद मेट, वो किस साइड में जाएगा? टी साइड. जो बाकी आएंगे
A side.
तो क्लियर हो गया आपको. चलो नेट प्रॉब्लम. चलो भी ठीक हो गया.
Ribosome combines with a small ribosomal subunit. The large subunit shows 3 sites: the acceptor site or the A site, the peptidyl site or the P site, the exit site or the E site.
Foi como super fone.
आजची नेट बराबर आज को नहीं यूज.
Não é problem, ela está lá o broadband.
It was like broadband. What?
Pata?
Connect, disconnect from what? Like
Não, eu mexi bütün kontrol ettiğim. Interesting bana, है ना?
bad luck. Yung rumah atas kan? Pati pa.
The exit site or the E site.
नेट चैनल છે ને સર
Can't connect to this net. Hey, we should have a message from Maglu back. Is something in network.
Flight mode is operating.
हम्म
Flight mode.
अच्छा ठीक है
Quickly net on. अब बस कनेक्ट हो.
Dial site or the P site, the exit site or the E site.
साइट है
This whole unit forms the initiation complex. This is followed by the elongation stage.
elongation second elongation
During this stage, another tRNA carrying molecule of an amino acid approaches the mRNA ribosome complex and
This
fits
side
in the A site.
A site very good देखो
Then
आप यहाँ
a
पर
bond is formed
देखो
between
bond
the
is formed that is bond is called a peptide bond
bond.
between amino acid first and amino acid second the CWH group of amino acid first and NH2 group of amino acid second they form the peptide bond ये peptide bond अतन में भाई तमिस कुछ लाइन पानस कुन सु कुन गैस
TRNA is S multivasan.
टीआरएन तो अब खाली हो जाएगा वो बाहर जाएगा आप देखो देखो क्या
threonine
ख्याल रखो
and the amino acid molecule on the tRNA.
यहाँ पर ख्याल रखो
As a result, met tRNA becomes deacylated. The ribosome then advances a distance of 1 codon and the deacylated
ये ribosome move
tRNA
करेगा।
shifts to the
देखो।
E site from where it dissociates.
ओ गया वो ले बाहर खाली टीआरएन
Meanwhile, another tRNA carrying an amino acid molecule attaches to the A site.
अब इस साइड पर फिर से आएगा
This is followed by the binding of
But you
the
have
amino
one.
acid
Peptide bond.
molecules. Repetition of this process leads to the formation
आई
of
बात
an
समझ
amino
राइबोसोम
acid
मेरा
chain.
मूव
Yes, sir.
This
Then they have
event
a
is
enzyme
called elongation.
the process will now continue
Finally, when the UAG codon or the stop codon reaches the A site, elongation is terminated.
terminated
Termination is the last stage of protein synthesis. The chain of amino acid molecules is released from the ribosome. This released amino acid chain is the protein, and this part of protein synthesis is known as translation.
translation this protein molecule
Then the tRNA detaches from the mRNA. Ribosome detaches and dissociates into its small and large subunits. Summary.
some
Protein synthesis shows that the first stage involves the binding of met tRNA to mRNA and the small subunit of the ribosome.
then larger
The
subunit
larger subunit of ribosome then combines with the small subunit. Second stage is the elongation stage. In this stage, the incoming amino acid tRNA fits in the A site. Then a bond is formed between methionine and the amino acid molecule on the tRNA. The process is repeated until a chain of amino acid molecules is formed. The last stage of protein synthesis is the termination stage. When the ribosome reaches the stop codon UAG, elongation stops and the newly formed amino acid chain, which is the protein macromolecule, detaches from the ribosome. Subsequently, ribosomal subunits along with the tRNA dissociate from the mRNA.
करो बस understand
In order for our bodies to function, we need to supply